Assalamu alaikum طلاب, and welcome to the third and last lecture of the histology of the urinary system. As with the previous lectures, for the ease of uploading of the lecture into the Google Classroom, we will discuss the lecture in two parts, and this is part 1. Just to recap with what we said earlier in the previous two lectures about the kidney. The kidney consists of outer cortex and inner medulla. And the most important part of the kidney, which is its functional and structural unit, is called the nephron. And as you can see here is the nephron that consists of two parts. This rounded structure is called the renal corpuscle, and the other part is a series of collecting tubules and collecting ducts that will transport the urine from the renal corpuscles to the outside. Here a histological slide of the cortex of the kidney, and as we can see we have a cross sections of the renal corpuscles surrounded by urinary space. So this is a glomerulus or renal corpuscle surrounded by Bowman capsule, and the space here is the urinary space where the ultrafiltrate will be filtered from the capillaries to the urinary space. All these structures in between are cross sections of the proximal and distal convoluted tubules, and if we increase the power of the microscope here we can see the distinct feature of each one. Here we have a medullary rays, which is a collection of striated tubules running through the cortex of the kidney. These consist of collecting duct in the center, and on each side thick descending part of lobe of Henle, or we call it proximal striate or distal striate tubules. They run within the medullary rays. Again, just a recap of the glomerulus, and as you can see here, this is the glomerular tuft of capillary that's surrounded by Bowman's capsule. Bowman's capsule consists of two layers, parietal layer and layer in intimate contact with the glomerular capillaries, which is called the visceral layer. The cells of the visceral layer is podocytes, and they are very characteristic cells, and they have a very important role in all kidney diseases. Each glomerulus have two poles, a vascular pole where an afferent arteriole and efferent arteriole enter and exit the glomerulus, and a tubular pole where the collected filtrate will be transported into the proximal convoluted tubule. The ultrafiltrate will then pass from the glomerulus to a series of ducts, and they are named according to their shape, whether they are convoluted or striate, and whether they are proximal to the glomerulus or distal. So we have proximal convoluted and distal convoluted, and we have proximal striate and distal striate tubules, and in addition we have the lobe of Henle. The renal corpuscle is almost always located within the renal cortex, surrounded by sections of the proximal convoluted and distal convoluted tubules. However, the medulla, we can see the proximal striate, distal striate, lobe of Henle, and other parts of the collecting system of the nephron. So here again we have this, our favorite photo of the nephron and its part, and as we said, we discussed earlier in the previous lectures the renal corpuscle, proximal convoluted, proximal striate, lobe of Henle, distal striate, distal convoluted, and now we will discuss the collecting tubule. The collecting tubule is not just a conduit for the transfer of urine from the distal convoluted tubule to the collecting duct and then to the pelvic calyceal system. It plays a very important role in the final concentration of urine, together of course with the collecting duct. Both of them, the collecting tubule and collecting duct, will concentrate the urine according to the need of the body, according to the state of hydration of the body. So if the body is in a dehydration state, the urine will be more concentrated by the action of collecting tubule and collecting duct, and vice versa. If we drank a lot of water, the urine will be diluted also by the action of cells of the collecting tubules and collecting duct. The collecting tubules and collecting ducts, basically they have the same lining epithelium, and these are lined by two cell types. We have the clear cells that are most abundant, and from their name they are lightly stained cuboidal cells with a few cytoplasmic organelles. As we can see, this is an electron microscope showing a clear cell in the wall of collecting tubule. These cells have basal enfolding, basement membrane enfolding are prominent in the proximal part, but this will become less evident and gradually disappear in the distal part of the collecting duct. These cells, the clear cells, have abundant water channel in their plasma membrane, which means that they have abundance of the protein aquaporin that is responsible for the transport of water from the lumen of the collecting duct, then it will be absorbed to the renal interstitium. The second type of cell is the dark cell, and from its name they are darker in color in histological sections. However, they are less in number than the clear cells. They are rich in cytoplasmic organelles, especially mitochondria, and as we can see here, all these cross sections are mitochondria, and they have a well-developed microvilli on their luminal surface, in contrast to the clear cells that have less well-developed microvilli on their luminal surface. And as we said, the clear cell, their function is the reabsorption of water from the lumen of the collecting duct. However, the function of the dark cell is to secrete bicarbonate or hydrogen ions into the lumen depending on the need of the body. The next part of the nephron after the collecting tubule is the collecting duct. The collecting duct, as you can see here, run vertically within the medullary rays into the medulla. All of the medullary collecting ducts merge near the papillae to form a large straight papillary duct that runs into the tip of the papillae. This is the tip of the papillae, and we can see here different collecting ducts are merging to form this large papillary duct. This collecting duct, as we said, is lined with the same type of cells that line the collecting tubule. So we have clear cells and dark cells, and the clear cells are the most prominent with a few dark cells. However, as the collecting duct pass deep in the medulla, the clear cells will become taller and more columnar, especially near the renal papillae, and gradually the dark cell will decrease in number until they completely disappear near the renal papillae. The collecting duct is the last site of the nephron where water absorption occurs, which is because also of the presence of aquaporin proteins in the wall of the clear cells. This is a histological section of the kidney, of the medulla, showing cross sections of two collecting ducts. And as you can see, the cells are large, cuboidal or columnar, with few microvilli on their luminal surface. Electron microscope features of the collecting duct show that these cells again are large cells with large nucleus, basally located, a few microvilli on their luminal surface, and they have a few cytoplasmic processes. This is the clear cells that line the collecting tubules and collecting duct. The collecting duct cells have receptors for a very important hormone, which is the antidiuretic hormone. This hormone is released from the posterior pituitary in response to dehydration or reduced water intake by the body. The action of these hormones on its receptor will make the collecting ducts more permeable to water, and by this it will increase the rate at which water molecules are pulled osmotically from the filtrate. The ADH antidiuretic hormone will bind to the receptor, and this will cause movement of the aquaporin protein from the inside of the cell, where it is stored as vesicles, to be bound to the luminal surface of the cell. That to say, it became a transmembrane protein. So it increased the number of water channels present in the luminal surface of the cell. These water channels will reabsorb water from the filtrate in a much faster and more efficient way, that we will conserve, our body will conserve water to end the dehydration state. Then the high osmolarity of the interstitium of the kidney will draw water passively from the collecting duct cells, and then this make the filtrate will as high concentrated material. The final part of our part 1 of our lecture is the renal interstitial cells. These are special types of cells located between the parts of the nephron that we have discussed earlier. And they are present, of course, in the cortex and in the medulla. So, for example, here we have cross sections of the collecting duct, and in between these collecting duct we have vasa recta and blood vessels, and also we have interstitial cells and interstitial matrix. These interstitial cells and interstitial matrix, or collectively known as interstitium, renal interstitium, is relatively less in concentration in the cortex, and it's estimated to constitute about only 7% of the volume of the cortex. However, as we move down into the medulla and near the papillae, the number of interstitial cells and interstitial matrix will constitute more than 20% of the volume of the medulla. While the renal interstitium in the cortex is very simple and basic, formed from fibroblast-like cells and few macrophages, in the medulla the interstitium is much more complex. It's composed of loose electron-lucent acellular material, which is the interstitial matrix, formed mainly from protein, glycosaminoglycans, collagen fibers, lipid droplets, and other materials. The cells, the interstitial cells in the medulla, resemble myofibroblasts more, in that to say they have actin filaments in their cytoplasm, and they also have a most characteristic lipid droplets and other cytoplasmic organelles and few mitochondria in their cytoplasm. The most important function of the interstitium, of renal interstitium, is the water and salt homeostasis. That's to say, its share in the regulation of water reabsorption and salt secretion into the filtrate, together with the epithelial cells and the endothelial cells of the renal capillary, that's to say the vasa recta, that is present around the collecting duct and collecting tubules. By this we reached the end of part 1 of our lecture, and see you in part 2. Thanks for listening.